Okay, so let's get into 15. Like I said, I want to get into as much of, well, all of 15, and then move into 16 as much as we can today, so that so I can kind of keep you guys on schedule. I don't want to rush through information, but I do want to make sure to get it done. There is a piece at the end I might optionally exclude. No big deal. It's more like a second example, but you'll get one example instead of two. But anyway, we'll talk about that later. Let's go into 15. In 15, we take what Mendel had discovered in 14, right? What Mendel had discovered with heredity with his pea plants, and we extend that to the idea of what was being passed down to the offspring, right? Now, Mendel was making an observation. He was noticing what was happening when he mixed two certain colors of plants, that they had certain ratios of traits. But of course, at the time, he didn't know what was being passed down. We didn't know about chromosomes. I was going to look up when Mendel lived. It was 18th century or something like that. You can do it on your end if you want to. I forgot to do it before class. But no doubt, it was well before we really had a good understanding of what heredity was, okay? What was it that was being passed down? As a matter of fact, the first thing they thought was being passed down was some kind of protein. That everybody understood the sex cells thing, but what the sex cells were bringing together was what was in question. They first thought it was a protein, but we'll talk more about that later. Come to find out, of course, it's a chromosome. This is a really cool picture of a chromosome. This is, again, what we can do in a modern microscopy is we can make different atoms of different types, make them glow, or we can replace, say, like we might replace the phosphorus atom that's in DNA with an isotope phosphorus, so it does the phosphorus thing, but because it's an isotope, it's radioactive, so it glows just a little tiny bit. The cool thing is we can make things that are hard to see more colorful. Like what you're looking at here is a locus, okay? So a spot on a chromosome. Remember, locus and location, that's why the words are really nice to think about. So that's a spot on the number 6 chromosome, okay? And so there's a number 6 maternal, number 6 paternal. I don't know which one this is, but that's one of them. So a spot on a chromosome is a gene, is it not? Right? Let's assume it's not always a gene. Remember I said some of the spots on your chromosomes aren't red, but just for sake of argument, what they've done here in this picture, they've actually caused a gene to fluoresce, the spot, the yellow spot. Now, what I want you to tell me, though, is what stage during cell division are these chromosomes? Are these chromosomes or chromatids? And explain if they're both. Like, why are there 2 dots is what I'm getting at here. The number 6 chromosome only has one gene, whatever color, the yellow one is coloring. Why in this picture are there 2 dots? Let's see if you all remember your mitosis. I'm going through. Yeah, there you go. Those are 2 tids, aren't those? Those are 2 chromatids. 1 chromosome, 2 chromatids. That's why there's 2 spots. We've made a copy of the chromosome. Isn't that a cool picture? So that's right there, physical evidence, right? We can actually see a gene, excuse me, a locus being passed from one to the other. All right? So very cool. All right, now let's turn our attention to what we're going to look in 15. One of the things that we learned about the chromosomal basis of inheritance, again, taking what Mendel's work did, whatever the verb I'm trying to find, and turn it into where does that happen in your body. It's really cool enough to watch an organism do something, fly, swim. It's much, much, much cooler to figure out how they do it, right? It's like the kid that takes the toy apart. It's not enough just to have the toy. You want to know how it works. We're all kind of that way, male or female. That's not a male or female trait. We all want to know how things work, right? And one of the things that we learn when we start investigating how genetics is chromosomal, how you look like and what you are is based on some chromosome, a molecule. One of the things we look at is sex, right? We all know that idea that females have 2 sex chromosomes that are the same. Both of them are X's. And if you're a male, you have 2 sex chromosomes. You have 2 chromosomes of everything, don't you? You always do. You have pairs of homologous chromosomes. So the sex chromosomes, while they're not the autosomes, remember the autosomes were the chromosomes found on the other chromosomes. And for us, we have 22 pairs of those and 1 pair of sex chromosomes. So if you're looking at this picture, isn't that neat? This is a real photograph too. All the little curly fuzzy stuff are loops of chromosome wrapped around a scaffolding or a protein. I'm sorry my voice is cracking. My wife has been complaining about her allergies for a few days, so I'm wondering if that's what I'm getting too. Of course, sometimes she thinks she's getting allergies, and before you know it, I'm sick. And I'm like, no, you had a cold. All right, but is this a male or female, by the way? Looking at this picture. Yeah, it's a male. See, the male and female, or the sex chromosomes, the X and the Y, aren't really truly homologous, are they? Remember, homologous chromosomes are identical pairs. They may not have identical information, but there's a number 6 paternal and number 6 maternal. In this case, identical, you know, in that phrase, identical, one of the things it implies is length and shape. Obviously, these two are completely different shapes. But I think I mentioned to you one time that one of the theories behind the Y chromosome is that it's a part of an X. There was a book written years ago called The Fragmented X. And what they did was talking about male characteristics, okay? So there is a part of the Y chromosome that can stick. So if you think about the Y chromosome as maybe being half an X, there's still parts that allow it to become homologous, let them stick together. Now, the interesting thing on the Y chromosome, there's something called an SRY gene. The SR is sex determining, okay? And this is the sex determining region of the gene. This is one of the spots. There's many different spots that contribute to your sexuality, but I know everybody gets disappointed, especially the males, when we talk about this in zoology course. One of the things we mention is there are primary and secondary sexual characteristics that all organisms have. Males and females have one primary sexual characteristic. Males have testes, which come from the SRY gene of the Y chromosome. Females have ovaries. All those things that we think about sex, all the external stuff, all the things we use are secondary sexual characteristics. And all the boys kind of hang their heads. Oh, really? My penis that I'm so enamored with is really nothing more than a secondary sexual characteristic? Yes. Your testes are the important thing. Anyway, so, all right. That's kind of cool, right? X and Y. Not everybody does it the same way, though. We do. A lot of us, we mammals, a lot of us animals do it this way. And this is so interesting when you think back about royal history. You know, the queen is supposed to give the king a male heir, right? And, of course, biologically, I wonder if they knew back then. I wonder if the scientists knew. Maybe not right back then. But anyway, biologically, among most critters, including ourselves, it is the sperm that determines the sex, right? Because if you look at the sex chromosomes, now the 44, those are the autosomes we're doing with, not pairs here, right? Plus the sex chromosomes. So males are XY, females are XX, and during meiosis, you're going to get 22 autosomes and one of the sex chromosomes, right? If you're male, though, you're going to have different sperm because you have two different sex chromosomes. But if you're female, you're only X. So that's why it's the male. So if it's the Y sperm that fertilizes the egg, you get a male. And if it's the X sperm that fertilizes the egg, you get a female. And I think it's, oh gosh, I think the fertilization rate in humans is something like about 50%. I think 50% of the time a male, 50% of the time it's a female. But I think there's a different mortality rate. Or is it the other way around? It might be a different fertility rate, but the mortality rate. So anyway, males and females end up, for the most part, similar in proportion, I believe. That might have changed. Who knows? Cooper left. Where'd you go, Cooper? Oh, computer went to sleep. There you go. I didn't realize that when y'all's computer goes to sleep, it leaves the session. All right. Anyway, so just some of the other critters, I don't really care. I'm not going to ask you on exams about these, but it's not the same in other critters. Like with insects, the sperm still determines the sex, but it's really not a matter of what kind of sperm. It's a matter of what kind of sex chromosome. It's whether you have one or not. That females get an X from the sperm, while males, having the X from the egg, get nothing from their sperm. As far as the sex chromosome, they get the autosomes, right? In chickens, it's the other way around, and egg actually determines the sex in this case, right? You can see in this case the females are different. They're ZY. You all know the female symbol, obviously, and the male symbol, they're the same. So it's an opposite over here. And then among some insects like bees, really interesting, like they have this haploid diploid system where males only have half the number of chromosomes. No laughing. Males have half the number of chromosomes, and females are the only ones that have a full set. If the egg is fertilized, so that's the determination of sex. It's not what you're fertilized with, it's whether you're fertilized. If the egg does get fertilized, what? If a woman is highly acidic, they all are highly acidic, and then you, depending on how you look at it, then you could kill off the fast males, and the slow, strong women's sperm make it to the egg. Maybe. Do Y sperm swim faster than X sperm? They do? Okay. So I get you what I said earlier about acidity. Yeah. So what we do, if you make more males, many of them die in the initial arrival at the party. So you have about maybe the equal proportion being able to do the fertilizing. I have to look into that. That sounds interesting. What, in male sperm live shorter than female sperm? They don't last as long? They don't last forever, yeah, okay. Hey, this is what I want to talk about now are some genes that are found, and that's really cool, Beth. I got to remember to look that up about there's all kinds of cool things about sperm. People have read entire books. You're like, really? But it's when you look into it, you're like, wow. You understand this whole evolutionary thing. Some genes that don't seem to really have anything to do with your sexual characteristics, whether they're primary or secondary, are found on X chromosomes. They're called X-linked genes. Now, it's kind of like it doesn't really happen this way because there's plenty of space. But it's almost like Mother Nature haphazardly maybe ran out of space and stuck these chromosomes on the sex chromosomes. Again, that's not really right because there's lots in your DNA that is junk space where they could have been put. Remember, folks, this has all happened randomly here, right? So for whatever reason, randomly, these X-linked genes, and I've given you an example of some of the characteristics, some of the phenotypes people have when they have these X-linked genes. You can be colorblind, Duchenne muscular dystrophy, hemophilia, right, the bleeding disease. This disease is very often found in which sex? Male or females often have these kinds of diseases. Color blindness is probably one you're most familiar with, unless you've done some, if you've done some study into the royal family, you realize that the high rates of hemophilia in the historical Roman families is because of folks inbreeding, really, but people who have this X-linked characteristic. I don't like this chart. I wanted to introduce you guys to the concept because this is the chart in your book, but the X's and the N's and the capital N's and the lowercase n's are very confusing to me. But there is something here I want you to note. These kinds of characteristics, these things that are not sexual, but genetic characteristics, as I said, characteristics don't have to be good, are quite clearly not good, right? Oftentimes what happens with genetic characteristics that aren't good, that are detrimental, they're recessive, all right? The idea is then, if you think about it and you do the Punnett square, if it's a really simple gene, in order to be or have that disease, you got to be homozygous recessive. You only got 1/4 of a chance of being that way. Anyway, you have a much less chance of having these kinds of diseases, obviously, but that gives you the general idea. Anyway, the whole idea here behind these kinds of diseases being more oftenly found in males is because males only have one X chromosome. See, they don't have two. They're XYs, right? Females are XXs. Now, these genes are on the X chromosome. All right. Let me go to the next page. I want you to simplify when you do these kinds of genetics problems with just using the letter C. All right, the lowercase letter of N in the previous slide indicate that these diseases are recessive. All right, so we're going to stick with a lowercase c. But instead of a bunch of C's and a bunch of N's, what I've done is if there's a C at the X, then you carry the colorblind trait. Okay, and if there's no C, then you don't. Colorblind is recessive. So, whether you carry the dominant allele or not, all right, if you're male, you're still going to be colorblind. See the male up there? He's got the colorblind allele for colorblindness. Now, let's say he's got the dominant allele. How come he's colorblind if the colorblind allele is recessive, or if the colorblind trait is recessive? I'm sorry, I keep saying allele. How come he's still colorblind? Regardless of whether he has a dominant or recessive allele. Remember the definition of a recessive allele. You might have to go back in your notes. You got to remember, well, that does happen. But remember, the definition of a recessive allele is it does not express itself unless it's by itself. So go back to that male at the top. Let's say that the colorblind allele he has is recessive. Why does he express it? Why is he affected? Why is he colored? Was it dark orange? Why is he colorblind? If that's the recessive allele, why is he still colorblind? Recessive alleles only express themselves when they're by themselves. There might be two of them, but they're by themselves. There's no dominant, because then you'd be heterozygous. Is that male up there does he only have one allele for colorblindness? Is there one on the Y? We're not talking about Y-linked genes here. We're talking about X-linked genes, aren't we? Why are males colorblind regardless of the allele they carry? Come on, fill in that sentence for me. Think about it. Why do you normally think about males when you think about someone, if you know someone who's got these diseases, or if you look it up? Does it matter which allele that male up at the top is carrying? If he's carrying a recessive allele, doesn't that mean he'll still, he doesn't have another X. See, but look at the male for a second. Hold on. He doesn't have another X. He has a Y. Now, it doesn't matter whether that little c is even a big C. We keep it as a little c just to make it consistent, because we remind you that it is a recessive disorder. It doesn't matter if you could say, well, there are dominant colorblind genes. It doesn't matter if that male up there, what allele he has, okay? In this particular case, it's more a question of absence or presence. Because he has the colorblind allele, whatever type it is, who cares? He's colorblind. On the other fact, in the female, she doesn't have a colorblind allele, all right? So now look down below. He's going to produce sperm that are perfectly normal. Therefore, there is a rule among colorblindness. Colorblind boys, or excuse me, the other way around, colorblind men, fathers, cannot make colorblind boys. Got it? See there in the chart, both those males are fine. They also cannot produce colorblind females. So if dad is colorblind and mom is perfectly normal, she's not like colorblind or anything like that at all, then they will produce children. None of them are colorblind. Some of the females, though, are in pink color, right? They're what we call carriers, right? They're carriers. As Raymond mentioned, carriers carry the genetic trait but don't exhibit it. So it's again kind of like being recessive, right? And being heterozygous. If you were capital P, little p, remember for the plants, you had the white allele, but because there was the dominant allele there, you were going to be purple. Same thing is going to happen here with that X chromosome. All right, now, as far as the carriers go, that's where you get a situation where colorblindness shows up. Let's go down below to the left-hand side first. Let's take a woman with normal vision. Dad was colorblind, but as far as she knows, she's not colorblind. Marries a guy, he's not colorblind, right? It would always be easy to tell if a guy's colorblind. On the other hand, her being a carrier, that disease is hidden. That condition is hidden, whether it's hemophilia or whatever. If we just change the letters, we could be talking about hemophilia if we change it to H. So along comes her mate. He produces Y sperm. All right. Now 1/4 of his offspring are going to be male with normal vision. But look where the colorblind trait comes in. And that's where the male says, or the father says, well, that's funny, I'm not colorblind. Why is my son? And the doctor says, well, because you gave your son the Y chromosome. Well, where did he get the colorblind? My wife is not colorblind. No, she isn't. But because it's a recessive disorder, she can hide it. She can carry it. That's what carriers do. Carriers don't. Now, you can go through the rest of the Punnett square and realize that even though they're going to, half of their females are going to be normal, while half of the females they have are also going to be carriers. But the important part is, you just do it in a Punnett square and figure it out. The important part is that's how we trace these X-linked genes. You know, that's how we go back through time and figure out where they came from, because we know they have to come down through the X chromosome. They can't come through the Y in the males, right? Now, the males can contribute to how that disease is spread by producing carrier females, okay? They can also, think about the one in the lower right. If everybody's colorblind, right? If the mom and the dad are colorblind, they can also make a normal-eyed individual. All right? No, I'm sorry, not if everybody's colorblind. If mom is a carrier. If mom is a carrier, they can produce a colorblind daughter, a colorblind son, a carrier daughter, and a normal vision son. Now, as I said before, if everybody's colorblind, if mom is colorblind, she's like this, and dad, he's colorblind, whole doggone family's colorblind. Everybody. You can put it in a Punnett square to convince yourself, but everybody's colorblind. So when you are working genetics problems that are X-linked or reading or thinking about them, remember, there's going to be a sexual difference in terms of how it shows up in gender, because it's only found on that chromosome that is a sex chromosome. It's only found on a sex chromosome, the X chromosome. All right? So I like to use the C's when you're doing your problems. And again, just think about it. If the C is there, that X is colorblind, or that X is carrying the colorblind trait. And if the C is not there, then it's not. So don't think of it as dominant recessive. I don't want you to get confused about that. Think about it, presence or absence. So these carrier females, they are carrying one of their X's that has it. The other one does not. I learned recently that carrier females for colorblindness are still colorblind, though. Isn't that neat? Go back to that for a second. That the reason why they don't know that, or they're not normally detected, is because it's a very slight form of colorblindness, just barely colorblind, kind of colorblind. So you need very sensitive tests to do that. And the reason why is, in a female with her X chromosomes, it's not good to have two X chromosomes. It makes too much of a makes too much of a gene product, right? Homeostasis, balance. I'm doing with my hands, but you can't see me. Doggone it. All right. So even in females, males, we don't have to worry about that. We have an X and a Y, but in females, their cells have an X and an X. Okay. So what is going to happen? Don't pay attention to the cat for right now. Is in every cell in a female's body, one of those X's, and it's random, folks, one of those X's is going to shrink down and become inactive. In other words, it's not going to operate. When it's time to read the chromosomes on the X chromosome, that little shrunk down inactive one is not going to be used. It's only going to read the chromosome information that's on the active X, all right? And that's going to be random in every cell. It's why we get calico cats. Calico cats, not tortoise hair, folks, but calico cats, the fur color, the all mixy, is because of the fact that women are really, females, women too, are really mosaics of cells. What was the word the other day? You said it, Tim, right? Chimera. Yeah, they're a chimera of cells. If each of the one being different based on whether their X is active or not. And there might be a patch of cells in which there's a bunch of this X that's active and a bunch where it's not. And that's how you're going to get the black and the orange fur, depending on which one is active or not, right? Some might be, the orange fur chromosome is active and some it's not. So you get that mix. Now, this also says one other thing about calicos, doesn't it? Calico cats calico cats. Which sex are they usually? No, not all. Not all. Ding, 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 ding. Not all. I got an exception for you, but you have to wait to see what it is. There is an exception to that, a very rare but very cool exception. But I'll get to that in just a few slides. But yes, they're usually female. By the way, that little X You can see it on the chromosome. They call it a bar body. Anyway, now back to my original. Why some why carrier females are really colorblind? Truthfully, because carrier females have this mix of which X chromosome. Sometimes in their cells, it's the X chromosome that has the colorblind trait that's active, and in some of their cells. Now we're talking specifically about the cells in their eyes, right, for telling color. And in some of those cells, it's the other X. So think about it as being in some cells, the good X is turned on, and in some cells, the bad X. So females that are carriers have a intermediate type of color blindness. I thought that was cool, you know? I wonder if we could answer lots of other things about females based on that. I wonder if that explains mood swings during menopause. I wonder if somehow we can control which ones are the active and the inactive. Maybe the good genes that produce the calming chemicals would be more released than the ones that make me go, my gosh, its head's going to spin around. Oh, good thing Millie's at work today. All right, I want to show you something here, but I want you to use what you know already to tell me what you think the answer should be. Oh, I meant to scratch it. Oh, yeah. I meant to scratch it while I was distracting you. Hopefully you didn't see it. Anyway, all right. What we're crossing up here, and don't get lost in the symbology that we're using, but wait, basically, and I'm going to make it really simple for you. What we're crossing here is this kind of critter, okay, with this kind of critter. So what we're doing is we're crossing an organism that is doubly heterozygous, one that is doubly homozygous recessive, okay? Now, you can put this in a Punnett square, and it'll work out exactly this way, but I don't, it's a good exercise for one thing, and I don't want to take time doing that, but the ratio that you should get out of that, and how would I know? The ratio is basically the ratio that you get from doing a test cross twice. Isn't that what you're doing? You're doing a test cross. You're just doing it twice. You're doing it 2 different traits. You're doing a dihybrid test cross, right? And you know that either one of those is going to give you equal proportions of whatever the trait is that you're coding for. So if it was purple flowers, so if we crossed a heterozygous purple with a white, we're going to get half purple and half white. Remember that demonstration? So based on all of that, the ratio down below, there's going to be 4 different ways. Since it's a dihybrid cross, then you can have flies that have the what are gray bodies with normal wings, flies that are gray with short wings, and the black ones are that way too. But based upon that, what do you think the ratio, the predicted ratio should be for the genes on all the different chromosomes? What's the ratio that comes out of a test cross? Just a monohybrid. I give you 200 offspring. That's what comes out of organisms that are both heterozygous. Remember Raymond and Tim? That's the genotypic ratio. There's one more, come on, in the monohybrid. You could do, that's the dihybrid. You can, a phenotypic. You can do it real quick in a Punnett square, folks. Get a piece of paper and real quick do that. Take the A or the B, I don't care, either one, and cross them in a Punnett square. Be a good exercise. How many are going to be one phenotype? How many are going to be the other? Thank you. Half and half, right? So if you're doing this, the same test cross twice, and of course now you're going to have 4 different variants, then the ratio should be what? The predicted one I scratched out. What numbers should you find in there? If once I erase all that, what should be the ratio in a dihybrid test cross? Not just 1-to-1, because there's not 2 variants. There's actually 4 variants, right? There you go. And it is, right? But it did not turn out that way in this particular situation with these particular traits. Why? Look at the results. The results actually came out more of a 50-50 kind of thing, right? Not even 50-50, more like a 100% or most we're on one chromosome than the other, right? Why did that happen? Okay, linkage. Remember I was talking about blonde hair and blue eyes? During synapses, genes that are close to each other sometimes tend to get inherited. Think about it as a unit, right? So that screws up the proportions. So you're only going to get the correct Mendelian proportions, the 3-to-1, all those ones that you guys were rattling off to me, when the genes are sufficiently far apart. So their chances of being linked are minimal. But the interesting thing about this in science, this is the scientific method, right? Observation, hypothesis. What does Mendel say? Mendel said it ought to be 1, 1, 1, 1. Results. It's not. Time to change our hypothesis. Oh, let's see. Maybe it has something to do with the distance between genes. There you go. And we solved it. Bing, bing, bing. We're brilliant. Okay, how do we get that trisomy 21? How do we get sometimes these, what we call a change in chromosomal number? Now, we're going to talk about chromosomal mutations. We're going to talk about first in number, and then we'll talk about in the actual chromosome being messed up. But this is a very interesting process that occurs. Let's start with the one that's occurring on the left. And we're going to be following an egg or a sperm. It does not matter. So the intent here is to figure out how did the chromosomes get screwed up. Let's go through meiosis 1. So I'm up on this cell right here. This is the question that I asked on the exam. I haven't finished grading all of them, but I made it through most of them. You know, the importance of where the kinetochore microtubules were attached. That's different in mitosis and meiosis because in one it separates those chromatids in mitosis, and in the other one what it does, sorry folks, and in the other one what it does is it separates first the homologous chromosomes, right, for the synapses or the crossing over points and all that, and then it separates apart. But anyway, what if there is no spindle fiber? You know, what if the spindle fiber didn't form? What if that M phase, or what if that mitotic spindle checkpoint that occurs during mitosis failed? And it does. It doesn't happen, you know, planes crash. Things happen. So do you see, do you see what's going on from the two pictures there? Do you understand why those two cells result from that? See, if those cells don't come apart, if they don't disjunct, right? That's a cool word, non-disjunction. Junction to come together. Disjunction means to not come together. And then you put the non in front, so it's not come together. So in other words, what it basically means, if once you do all the math behind the word there, is you realize that they didn't come apart. They should have, but they didn't because there's no spindle fiber. Somehow that checkpoint got messed up. Somehow it got slipped through. So what we end up doing for that set of homologous chromosomes, all of them end up in one cell. So now during the number of chromosomes are divided in half, but in the other chromosomes. So we're just following this one set down below. So that one set that didn't disjunct, that's all these down here now. And you notice there's none of them over there. So the problem is that when they divides again, right, when it divides in meiosis 2, what you're going to end up with is half of your cells will have an extra chromosome and half of your cells will be missing a chromosome. That's not such a good thing. You're supposed to have the normal set of chromosomes is 23 from each parent. So what we're saying here is that this kind of non-disjunction, this one that happens up in meiosis 1, when that happens, the parent, for those particular gametes that are produced, half of them will have too many chromosomes and half of them will have too few. Now, there are problems with having too many or too few, but as far as having too many, if that extra chromosome, see those two long ones, what if that was the 21st chromosome? And then the male sperm was, had just carried one like it's supposed to. Well, if there's 2 in the egg and 1 in the sperm, now you got 3. That's Down syndrome, right? We'll talk a little more about, these kinds of things. So if a non-disjunction is going to happen, if it does happen, then it's better that it happens in meiosis 2, though. See, if the homologous chromosomes separate correctly in meiosis 1, then the number thing is not taken care of completely, but its chances are cut in half. See what I mean by that? The non-disjunction occurs during anaphase 2, if the spindle fiber again is missing, then half of the cells are messed up and half are not, right? Specifically, a quarter of them have too many, quarter have too few, but the other half are fine. So a non-disjunction is not something that's good no matter where it occurs, right? Obviously increases the chances of a change in the chromosome number, but I guess if it's going to occur, that the odds of the problems, of course, are going to be much less if it occurs during meiosis II. Isn't that neat? It's not neat for the folks that end up, the product of those gametes. Don't get that confused now, folks. We're not just making cells here. We're making gametes. We're making special type of cells. Now, there is a process that we'll talk about in the next slide, too, whereby this happens to all of them. So the non-disjunction is all down the lines. Very odd, very odd. All right, so a couple of terms here. If you're missing a chromosome, you have one too few, or the organism does, then it's considered an aneuploidy. So I put the math in there. Doesn't matter what species we're talking about. It's all about the missing or have too many. And then if you produce diploid sperm, which does happen, get this complete failure non-disjunction that occurs, or complete ovum. Ova is plural, ovum is singular, sperm and sperms, yeah, I guess, ova and ovum. You can actually end up with an organism that's what we call tetraploid. So haploid, diploid, triploid. Got ahead of myself. And then tetraploid. Tetra means four, right? Tetrads. A tetrahedron has four sides. Now, do these organisms survive? No. Well, I shouldn't say that. Are these organisms, I'm going to introduce you to a term. In science, we never like to use the word really normal. We'd like to use the word wild type. Wild type is normal. It's the way it usually is. So are these organisms going to be the same as the wild type? No, they're not going to be the same as us two N individuals. Someone with an extra 21st chromosome obviously suffers conditions, and there are other things that are going to cause problems here. But then again, when you get to this point, that's a real problem. Now, among animals, this is not very common. And among animals, this does not happen very often, although it does sometimes happen. But plants, for reasons that I'll talk about if you want to take my Bio 2 class, plants seem to be a little more tolerant of it for some reason. The reason why I bring that up is a lot of the stuff we eat, corn and all that sort of stuff. We were talking the other day in class about GMOs, but wheat is a descendant of a wild grass that we artificially stimulated to engage in this all of them, this polyploidy process. That the modern wheat that we eat today is a derivative of this naturally growing grass that now has just a bunch more chromosomes. Remember I told you the other day that watermelons had like thousands of chromosomes or something like that? But if you're like this unfortunate child, then you can see in her karyotype that right here, I can say her, of course, because she looks like a female, number one, and right there. And then, of course, there's the problem. About 1 in every 700 children, which is higher than I would have thought, but it has gone up down below as the mother gets older, but not really understood why. Obviously, the age of the egg is a concern, but we're not sure exactly the mechanism. Because remember what happens in this with a female, she's born with all of her eggs as meiosis I. It's only when the egg is fertilized that it undergoes meiosis II. So it's kind of like, is the mess up at the beginning, the non-disjunction? Is the mess up after the sperm fertilizes it? Because the cell or the egg doesn't undergo meiosis II till it's penetrated by the sperm, right? During your menstrual cycle for females, what they get rid of is a meiosis I egg. It doesn't. Why go through meiosis II if the thing is, why put on lipstick if you ain't going to the party? That's a horrible analogy, but you understand the point. Why go through all that effort and nothing going to happen? So if it's when the sperm fertilizes the egg, then how much maybe is the sperm involved? There is some research that shows as males get older, they continue to produce sperm. We can always inseminate, but the process remember when sperm aren't used, they get reabsorbed, but the process by which we do it starts screwing up. And even males can actually contribute to trisomy 21. So it could be the sperm that has the extra chromosome. Not as often, apparently, but in females, much more often, yeah. I think the thing to me is a parent Down syndrome. The kids, when they're first born, of course, the idea of the disease is not real apparent phenotypically. The typical phenotype that we associate with them just is more apparent as they get older. So I wonder from the parents' point of view how heartbreaking that must be to say, oh, the child looks fine, and then, not to say anything that you couldn't love a child like that. I know plenty of folks with Down syndrome that were, and there's different levels of it too. Sometimes folks that have those kinds of Challenges and disabilities have these gifts in other places though too, so that's the cool thing about it. Okay, you can have aneuploidies to your sex chromosome. So here's the normal or wild. There you go. I should have used the word wild type there. But there's if you have it, and this works the same way. You have these non-disjunctions, so you can end up with an extra one. And if you're a male, by the way, if you have a Y chromosome, you're going to be male. You're going to be male on the outside. Now, you might be Klinefelters, so you might have ended up with 2 X's, okay? But you're still going to be male. Now, some typical phenotypes is there's a shrinkage in the testicle region, there's an expansion in the breast region. But, you know, there's a lot of guys out there I know who probably could use a bro. That doesn't mean they're Klinefelters, okay? So, but it seems among the sex chromosome aneuploidies that it's a little more received by the organism. In other words, the detrimental effects aren't as much. And I'm not a doctor. Klinefelters was probably named after a doctor, by the way. But I'm not a doctor. But from what I understand about Klinefelters, you probably maybe have met one. It's not so uncommon that you didn't, and you probably would have never known it. Now, the double Y one's kind of cool. There's this part of oh gosh, what is it, biology or sociology, called sociobiology. It's kind of cool to read some of these books. There was one by, I don't remember the fellow's name, Desmond Morris, I think, called The Naked Ape. It's real thin. You gotta pick it up. It's very cool. It basically is kind of he explains why we do things from our primate origins. He gets a little carried away in some places, but I do like the book overall. Like the one that I thought was kind of funny was Why Women Wear Lipstick. Red lipstick, right? According to him, the reason why that, what would you call it, custom, why that would have evolved is because out of our primate origins, when a female primate is in heat, they don't reproduce all year round like we do necessarily. The genital region will become engorged with blood. It become redder and larger. Okay, so he argues that that's why women put on lipstick. I've seen lots of women walking down the street with lipstick, and I don't think sex was what came to mind. So I kind of take that one with a little bit of a grain of salt, but it's some cool stuff. So anyway, there were some folks who took that and kind of applied it to the double Y. If aggression is part of the Y chromosome, if males are more aggressive because of some genetic aspect to it, not because your father used to beat the snot out of you when you were younger, but we would expect to find more males incarcerated for violent crimes that were double Ys than in the normal population. And I don't think it played out that way. I don't think the study supported or refuted that hypothesis. So I think that the results were inconclusive. Females, if the sperm comes in without a chromosome, which can happen because the other one has too many, you can get a Turner syndrome female. Again, traits aren't obvious from what I understand, perhaps a little stockiness to the body. And what I've been described as a webbed neck, which I don't know exactly what that means, but I've looked at some pictures online just to see. But, you know, if that's a neck, then I guess the skin's just a little wider, I suppose. It's not like the flying nun or anything like that. But again, you may have met folks like that and just didn't know it. Metafemales have menstrual irregularities. I think that's really the big, they probably have other. Females are so finely tuned that I'm thinking if you miss or have an extra chromosome, you probably have some hormonal issues in general. But one of the things I hear about metafemales is menstrual irregularities. But I want to caution females out there before you start freaking out. There are 1,000 things that cause menstrual irregularities, okay, 1,000,000 things. So just because you're having menstrual irregularities, whatever that might be, it doesn't mean you're metafemale or Turner syndrome, okay? Probably just means you're, you know, that fine-tuned machine just doesn't always work the right way. All right, you can do the Punnett square if you like, but I've talked about it as we went through it. But yes, this is the Punnett square that shows how this ends up. I do want to show you one thing, though. The Y sperm does nothing. So if the egg doesn't come in with at least an X chromosome, so you get a Turner syndrome, the Y, if neither one of them contribute an X chromosome, then you get nothing. You get non-viable. Doesn't grow, doesn't live, doesn't survive. So this is a karyotype. You see where I've circled the YY? This is how they would do that. Old days. This is the old days, folks. This is cutting it out of a photograph of the cells in mitosis, cutting out with a piece of paper with scissors and then pasting back together on a page. That's how I did genetics. With a candle under a bridge while it was raining and there were dinosaurs everywhere. This is a polyploidy rat, so this is one of the exceptions to the rule. It lives in South America somewhere. It's similar to the rats around it, but it's a lot bigger. So this is one of the animals that seem to be able to get away with this idea of just adding the number of chromosomes. That usually is a nail in the coffin when you start monkeying with it. You know, we talk about changing one letter. You know, we're changing the whole number here now. All right, any questions on changes to number? Pretty simple, right? A few new terms, but other than that, no big deal. This is not a hard chapter. Everybody there? Hello. Just want to make sure everybody's still awake. Cooper? Oh, no, Cooper's sleeping. I keep thinking, like, oh, I have 5 people today until I forget one of them was my dog. All right, let's keep going on then. Now, we can also have changes to chromosomal structure, not just chromosomal. So you can have the right number, but, and these are so simple to understand. Let's imagine the letters are loci. Now, they could be genes, it might be not be genes, whatever, I don't care. So keep in mind, well, I do care actually. Keep in mind that if, for example, like a deletion occurs, let's start with the first one. Let's say a deletion occurs of loci D. But let's say loci D, I might as well introduce you to the terms now. Let's say loci D was an intron. Okay, I wrote it out for you. The other one is exon. Intron is the gene intervals. I'm sorry. Yes, the gene intervals. And the exon is the expressed genes. You see what I'm saying? So the expressed genes are mixed in with the gene intervals. So it's a little bit confusing. We look at the word intron, you think intron included? No. Intron is not included. Think of the other way down. Exon expressed. All right, so let's imagine D is an intron. Let's imagine that what happened, some mutation occurred during chromosomal replication, which we'll talk about later on. D didn't get copied. So would that be a problem for that organism down below? Would that be a problem for the organism that's missing D if D were an intron? Why? What do introns do? Cooper, how about you? I'll type it for him. Introns are gene intervals. You're making it too hard. Making it too hard. Where's my phone ringing? My iPad. I'm sorry if that's bothersome in the background. Exons are expressed. So I just told you that D is an intron. It's a part that's not used. It's a part that's not expressed. It's not getting that piece of junk mail. In other words, the Marco's Pizza ad that gets delivered to my mailbox a couple times a week, and we only eat there maybe once every 6 months. If 1 week they miss it, am I going to miss it? Am I going to go, oh man, where's the Marco's? You probably am, because I wanted pizza that day. No, so if D is an intron, who cares? Now, if D is an exon, we've got a different story, right? How about if it's duplicated? Is that a problem? Yes, that's a problem if it's an exon. Why is it a problem? For what Beth Andrew Lindsay said earlier, everything should be balanced, right? That's a complete violation of homeostasis there. See, these aren't hard to understand, are they? This is good because I don't feel like I'm rushing through it, and now I'm going to have plenty of time to get into 16. All right, if it's in there backwards, then that's a mess up, because guess what? Your genes are just the way you read a book, left to right. Just because you got the question, you answer a question, you go to bed, Cooper. All right, so obviously if it's in there backwards, then that's a problem as well. Does your cell know it's in there backwards? No. Now, there are things that come along that try to fix these mistakes, but when it's time to read the chromosome and it's time to figure out what the code is saying, it's not going to know it's in there backwards. So guess what that's going to do, right? Let's make that connection way back to the beginning of the semester. So if the code's in there backwards, what is that going to affect? What is that going to change? What is that going to make wrong? It doesn't necessarily have to be wrong, but what is that going to change? Ooh, come on, you can do it, guys. Come on, Raymond. Come on, Beth. Come on, Tim. Think about it. So if the code is in there backwards, that code is going to change something. What's it going to change? Be afraid. Come on. It's back chapter, the protein chapter. Come on. Can't get any closer than that. Something to do with a protein. Thank you, Cooper. There you go. Nice, right? So this code is messed up. That code is messed up. That code is messed up. The secondary level is messed up. The secondary level is messed up. Blah-ba-dee-ba-dee-ba-dee-ba. Get the idea? In the end, boom, 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 the structure of the protein is messed up, isn't it? Right? All right, good deal. I love it. We're making connections. Sweet, sweet, sweet. Thank you, Tim. Thank you, Tim. Thank you. Awesome, awesome. Now, look at D. D's a complete screw up, isn't it? Now, here's what happened during pairing up in synapses. Somehow, what does my wife always say in that horrible way when she sees like a very attractive female and a male not so attractive? Mismatch, right? So what happened here is non-homologous chromosomes lined up. Why? Oh, I don't know. Because they just, they were drunk and they didn't know where they were doing. But anyway, it happens. It happens. But then they went through synapses. Oh my God, make it worse. And then look what happened. Now, does your body know that loci M, N, and O is now over on a different chromosome? No. And then let's say AB was not put at the end of the chromosome. What if it was put between the P and the Q? So if it said P, A, B, Q, R now. And what if the order in which that gene product was made was important? What if you got to make a P and then you got to make Q? But no, you're making P, then you're making A and B, and A and B has nothing to do with P or Q. So the problem gets mad worse as it's, yeah, heck yeah. You don't want a translocation, folks. You hope that cell dies off. You hope that cell goes, ooh, here's a picture of one. Do you see do you spot the translocation? See, do you love this color microscopic fluorescent microscopy? Oh, I always get that screwed up. Do you see how they highlighted the translocation for you? It's obvious, isn't it? Isn't this neat? You can look under a microscope if this was, and you go, oh, there it is, right there. That'd be so neat. I love it when I do that in science sometimes. When I go out in the wild and I'll identify some new species or I'll see something happening, I'm like, wow, that's so neat to watch it occur. See the difference in the colors, right? Now, the one on the red is not blue, but it's bluish green. Don't worry about this red over here. This is probably just what we call an artifact. That artifact is what you show me on the microscope slide all the time when you think you found something interesting. And I say, no, that's just a booger. Just a dirty part of the slide or something. Are we done with this translocation? Everybody understood it, right? That's a real screw up. Oh, chapter 16. Oh, boy. Another one. Man, I should just teach a class on this stuff. If there is an extra piece on the end, can it? Oh, so 2 recordings? Is that what you're saying? So do this one and then make a separate one for the part I go over? I may not have to, though. But is that what you mean? Oh, I get you. To be something in, can't it? No. It doesn't know that. These are molecules. It'd have to have some way to know that. Yeah, that's probably not going to happen. There are ways in which things like that happen, but in this particular case, no. Okay? But I'll show you about some ways in which it does happen. Okay, so now that we understood this whole idea of, what did I call the last chapter? I tried to title these chapters in such a way so it made sense. So the chromosomal basis of inheritance. Yeah, that's right. So now we're at the molecular basis of inheritance. So we're drilling down, right? That's what we're doing in these chapters. First, we started with Mendel's work, very open, very broad, right? Very much at the macroscopic level, right? And we're getting a little bit smaller. Now we're moving into the DNA, right? Everybody's familiar with this molecule. I love it. They discovered it in 1952. Watson and Crick, whom we'll talk about. And everybody's seen it a million times. I could show it to 3rd graders, and they know what it looks like. Isn't that neat, though? If I was teaching this class, say 70 or 80 years ago, we would probably just be questioning what it looks like. But it does. It looks like a ladder that's been twisted, right? It's what we call a double helix. A helix is a spring, right? A spring is a helix. Many species of snails their genus is helix. Anything that's winding around. The reason why we call it a double helix is because it's two things winding around. Right, so it's two rails of the ladder, and then the bases are matching up in between across the middle. These are making the rails of the ladder. How are they matching up? They're matching up like this, because that's the way they bond chemically. Now, this is just a much more sophisticated model on the right-hand side. Okay, so let me get this going here. There we go. All right, let's talk about some people who were involved in all of this. Miescher, 1869, he was the fellow that we normally credit with discovering the nucleic acids. Okay, you see, prior to this time, our thoughts about where heredity was, we thought it was a protein. We thought that your parents passed on some protein to you, right? And we understood the sperm and the egg, don't get me wrong. But we were saying what in the sperm and the egg is being passed on. We didn't know there were chromosomes. We didn't know any of that. Now, microscopes are getting better and better. And what Miescher did is we actually was able to describe the nuclear contents. So he was basically able to give the scientific community a place to look. He's like, okay, guys, I'm thinking this is where we ought to focus our attention. Because he probably was able to witness some of the cells undergoing mitosis. So he's probably like, let's see, they're dividing. Each one needs one. Maybe that's the recipe book. Can you imagine what he must have felt like too? God, all scientists, all their lives dream of that one time when you go, oh, bingo, eureka. The guy running down the street naked. Even Darwin talked about it when he was laying in bed reading. Well, actually, he didn't talk about it. I wonder if he did that. He's laying in bed reading something, and anyway, long story. But maybe Miescher did that. Maybe, but he gave us a place to look, right? And one of the first things they did, I don't talk about it. It's not on the slide here. But one of the first things they did is they said, oh, he's found where the protein is. Now, it's not a protein. I know, I'm not trying to confuse you. But he's found where the protein is. So you know what they did? Very simple. They took a cell, they took the nucleus, and on that cell, they treated it with this kind of an enzyme. Now, what do you think that enzyme did? It probably had a fancier name than that, but that's kind of a general name for what this ASE is, an enzyme. But what do you think the substrate of that enzyme was? So what would that enzyme eat, maybe? Yeah, there you go. So guess what? Think about it, right? If this stuff in the middle is a protein, then after treatment with the proteinase enzymes, come on guys, critical analysis. I know you all can do this too. When I look in the nucleus, what should I see? If that is a protein, hypothesis, the stuff in the middle is a protein, experiment, treat it with a proteinase enzyme. So you're looking at the result. With nothing. There you go. Nice, Tim. Thank you. Nothing. Didn't happen, though. Revised hypothesis. This is obviously not a protein. Still don't know what it is, but it's not a protein, right? We're starting to learn what it is. So we call Miescher basically the discoverer because he's the one who basically got us off the path of proteins. He said, okay, fine. I know where it's happening. I bet it's happening in this little spot right here, okay? Genetics, because I'm seeing copying and mitosis, so there's something about passing on something or other. There's the molecular part of it, okay? So they try with a proteinase. Okay, it's not that. Now, staining things in microscopes is really important. I've showed you some awesome pictures this semester. So in the early 1900s, a guy named Fulgen came up with a stain that stained these things in the nucleus red, these nucleic acids. He stained them red. And that made it nice, right? Like when we were looking at the mitosis and meiosis slides yesterday. It's nice. If those things weren't stained, good luck seeing the chromosomes. Sometimes you can, but it's a lot harder. He came up with a stain. We actually call it a Fulgen stain, and it stained nucleic acids red. But he's known for more than just that. Here's some observations he made. He was able to find out that somatic cells, right? The regular cells, not the sex cells. Somatic cells in all species were equal. You understand what that means? In all species, in the somatic cells, the nucleic acid number, the concentration, the amount was the same. Does that make sense? I mean, what I'm trying to say is, can you make the leap to what we know now and say, oh, this is when they started understanding that chromosomal number. Okay, how many chromosomes do I have in my nucleus? I'm a human. How many do I have? That's good. That's fine. We can do more than once. Thank you, Tim. Thank you. Thank you. How many do you have? How many do you have? How many chromosomes do you have? That's our polyploidy. How many chromosomes does Tim's identical twin have? No, Tim's identical twin. Tim's got, what, 500,000? There you go, right? You get the idea? So, of course, he found out that the chromosome number was the same. Now, look, take this to the next step. Guess what he found in the gametes as far as the amount of as far as the amount of stuff. So don't give me a number. Give me a fraction might help, or a proportion. How much stuff did he find in the gametes of those species compared to what he found in the somatic cells? Yeah. Right? Isn't that neat? So we're getting on this idea. Now, that makes sense. If I'm a scientist, I'm going, oh, look at this. This is more support of Mischer saying things are being divided in half. Oh, that's why everybody on Earth has got the same amount of red stuff, same amount of stained stuff, because every time we reproduce, we divide it in half, and then you two halves get together and we make a whole again. Ooh, bacterial transformation. Spooky, spooky, spooky. Although I wish I could love it. I would love if I could do this. Yep, genome. Yep, the Human Genome Project. We know where all the genes are. We know where all the loci of the genes are. We don't necessarily know how they work together or what they do, but we know where they all are. We're finding out that genes work with other genes and in conjunction with genes to control other genes, and it's a bureaucracy. It's almost like bureaucracy is genetics. Griffith, not Andy Griffith from a show that I love to watch. Andy Griffith always reminds me of my father. Anyway, here's what he did. Not so nice on the mice. Not so nice on the mice. Griffith was an experimenter in the 30s, and what he was looking at was this idea that whatever the genetic molecule was, now that we know that it's some sort of nucleic acid, he was kind of answering the question about its ability to store information, right? We're like heredity, how this all happens. So here's what he does. He takes a strain of, was it staph? No, Streptococcus. Streptococcus, I'm going to write it out for you. Hold on. There we go. Streptococcus pneumonia. It's a prokaryote. It's a bug. It's a germ. Okay. If you took Bio 2, you'd learn right away that Streptococcus means that it's actually a bunch of round ones on a strap, straight. Streptococcus tells us it exists. But anyway, the pneumonia part tells you what disease it gives you, right? So anyway, there are two strains of this Streptococcus. Yeah, I don't know if that's pneumonia. That might be another one. But Streptococcus that causes streptococcus is a Streptococcus. Yeah, in that group. But there are two strains of this Streptococcus pneumonia. There's, I always get them mixed up. There's the S strain and the R strain, okay? Oh, there we go. S strain is pathogenic, so it'll kill you. The or virulent is another word, by the way. The S strain I'm sorry. The R strain is non-virulent, so it won't kill you. It's kind of like we talked about with E. coli, right? It's the strain of E. coli that's the problem. You have E. coli in you, but you're not sick, okay? Okay, so here's what he does. Let's follow through. In the very beginning, he takes some living S and he injects that into the mouse, and there's the picture of it. Streptococcus is wrapped around by a capsule, that orange thing, and the reason why the mouse dies is because the mouse's immune system can't find the organism to kill it because it's wrapped in that capsule. Mouse dies, okay? He injects the living R, it doesn't have the capsule, the mouse's immune system does its job, boom, everything's cool. He takes the living S and he kills it, so he gets rid of the capsule, and he injects it into the mouse. Now, the reason why the mouse lives is because the S needs to be alive to do the killing. This is a control, folks. Sometimes organisms die not because of anything in the thing that infected them, but because of their response. What am I talking about? Give me an example of where someone would die because their body, say, over-responded to something. something that shouldn't kill you at all, but your body just went haywire. This time of the year, if you are someone who deals with this, you know what I'm sure, right? Bee stings. Why? What about the bee sting? I mean, bee stings hurt. They don't feel good, but they generally aren't going to kill you unless, right? Well, shock, you could be put into shock. Well, what I'm saying is, yeah, you got to be allergic, right? So you got to be allergic to that stuff. So did you die from the venom in the bee sting? No, it didn't feel good, but it shouldn't kill you. But if you are allergic to it, your body goes crazy, puts you in what we call anaphylactic shock. Your throat starts closing up. You can't breathe and you die. That's how those poor unfortunate folks die from peanut butter. Even the smell, like on a plane, can put them into that. It's real. I mean, I understand that. I don't want to die just because I'm allergic to peanut butter. I guess walk around with a suit. I don't know. What would you do without PB&J? Anyway, last part of the experiment. Here's the information part. What he does is he mixes together the S that is killed. Now, it can't kill the mouse, right, because the capsule's gone. And he mixes together living R, and that can't kill the mouse because it doesn't have a capsule. But the mouse dies. Oh, crap. Now, he cuts the mouse open. Oh, by the way, one of my pet peeves. He does not do an autopsy on the mouse. He does a necropsy. An autopsy would only be possible if he were a mouse. Auto, necro, dead. You don't autopsy a whale, you necropsy a whale. Unless it's a whale doing it. Okay, so he autopsies. Oh, shit, I just did it. Jeez Louise. All right, so he necropsies this mouse, and he finds that in that mouse, there's some living S. Well, he didn't inject living S to it. Well, here's what happened. The information in the heat killed, kind of. There you go. Not the S's genetic information. See, bacteria can do this thing called bacterial transformation. Bacteria that are living can actually take up DNA from the environment, incorporate it into their own. So what the living R did is it incorporated the DNA from the S, which allowed it to make a capsule. Mouse dies. I want to be able to do this. I'm going to take some bird DNA and fly. But I want to be able to give it back. And then I want to take some shark DNA and swim. You know, those kinds of things. I think that'd be neat. This is where my wife usually says, you need help. You can understand what he was doing here? He was showing us that it was information. This was good from the strep? What do you mean? I guess it was good for the... Yeah, it's a way that... Yeah, it's a way that prokaryotic organisms can adapt to their environment very quickly. Many times what happens, if you're not born with an advantageous variation, you can run fast or whatever, you're screwed, right? Among bacteria, it's akin to, let's say I can run really fast, but you can't. I could go to you, we connect each other, we switch some DNA, and guess what? Now you can run fast. It's why these doggone things are such a pain in hospitals. Staph aureus infections and all that sort of stuff, they don't have to go through this reproductive thing, which they do very quickly too, but they can just pass that stuff back and forth. Tetanus can do that. The thing that gives you tetanus can actually come up with against a tetanus one that can't hurt you, and then give it genetic information that it can. So yeah, kind of spooky in a way. More in bio too, but that's how they generate variability, or one of the ways they do it. See, they don't have sex like we do, so they don't get the crossing over part. Information, right? Info. Now these folks start working on the structure. So we're moving up into the 40s and the 50s. So a guy named Chargaff, and I'm going to refer to this a million times. I'm going to call it Chargaff's rule, and that's what Chargaff's rule says. The brackets around things means concentration of, numbers of, proportion of, whatever you want to say. But it's always A to T, and it's always G to C. And the reason why that is because there was a rule that fell out of the sky that says that, no, that's not the way it was. It was because of chemistry. A binds to T because it's 2 hydrogen bonds, and G binds to C because there's 3. It's like a key fitting in a hole. Now, sometimes do they accidentally bind messed up? Yep. My old pickup truck in high school, I could start with a screwdriver. I didn't need a key. So sometimes things get worn out, and they'll just bind up with anything. Now, this is going to be known as Chargaff's rule, ATGC. So let's do a little exercise in Chargaff's rule. You are in the lab one night, and you do a DNA analysis. And on that DNA analysis, the results you get are the proportion of adenines. You somehow have figured out a chemical process by which you can do that. And you find that in this sequence of DNA, 15% of the molecule is adenine. Here's what I want you to tell me. How much is thymine, how much is guanine, and how much is cytosine? I'll give you a second to do that. Okay, so you're saying that's how much thymine, Beth? Beth and or Lindsay? Okay, keep going. Close. No. Give me, let's do, okay. How much does A and T add up to percentage-wise? Okay, and then we still have two more bases to account for. We know they're going to be even. A equals T, G equals C. So then how much is G and C? Did you say, you said 35 before, didn't you? Oh, God, yes. All right, so we got 30 here. Got to have 70 here. We looked at the whole molecule. Now, each one of those can't be 70. Yeah, I'm sorry, Tim. The doggone numbers thing again. See how that works? Isn't that neat? From one piece of information. If you just remember Chargaff's rule, which I didn't, obviously, but it does have to add up to 100. So if I give you the concentration of one, it's the concentration of its complement, and then that's the concentration of those two. Take the other two, divide by 2, you've got your answer. Piece of cake, right? I love it. It's simple. Your entire genetic code driven by these 4 letters linked together in certain orders. Just amazes me. Wilkins and Franklin. Oh, gosh, I could go on this for a while. Wilkins was a female, brilliant female scientist back in the time in which female scientists weren't looked at very credibly or with respect. Her supervisor, I guess, is historically quoted as saying on one of her evals that she's brilliant, will do wonders for science, but she should be home having children. Anyway, the poor woman died young from ovarian cancer, but this is her, Rosalind Franklin. She worked with a fella named Maurice Wilkins, who really wasn't such a nice fella anyway, but that's another story. Anyway, they were taking x-rays of the DNA molecule. This is a picture of the x-ray of the DNA molecule, and if you're kind of going, what the heck? I know a couple of times when your doctor shows you x-rays, you're like, what is that? Here, let me see. See if I can help you visualize that. Took me the longest time. It's the Xing here. That's what you're seeing. See the Xing? See, if we look at the double helix from the side, X's are being made, right? Everybody cool with that? So keep that in your head. There we go. See them? The X's. So the DNA molecule is being looked at from the side. If I could do a double helix without making this a complete wreck. But you get the idea, right? Please tell me yes. So that's what he's looking at, or what they were looking at. Now, the interesting thing about when they were taking these x-rays isn't, well, the X thing's interesting too. It gives a lot about structure. Trying to figure out what this thing looks like. Is the hits on the x-ray, the exposures on the x-ray film, these little black dots, what they were finding is that there were certain numbers that always seemed to basically repeat themselves every time they took a picture of DNA, no matter from what critter or where. And I'll explain the numbers later, but I want to give them to you now. They noticed that this was a repeating number, a repeating distance. They're measuring things, folks. 3.4 nanometers, so we're down at the molecular level, real small. 1 nanometer, and then the other one was 0.34 nanometers. So make a note of that in your notes, because I'm going to talk about that. This, again, what we're doing is building what the DNA molecule looks like, so we need to know what's in it, and then, of course, how to arrange it. It's like building a house, right? Make one more observation now. Notice that these are related in some way, aren't they? Mathematically, they are. 1 times 10 is the other. Where are we? 17. Yeah, we're doing good. Okay, so we take all these guys. Let me go back to that slide again. We take all these guys that work. We take Miescher's work and helping us understand what it is and where it is. Fulgen's work for getting that splitting thing kind of into the conversation. Griffith, that it stores information. And then these guys down below, they're taking a lot of these guys' observations. It's all like an onion, building on top. and they're contributing. And Watson and Crick, who did not like each other whatsoever, were able to overcome their differences and won the Nobel Prize and figured out what DNA looks like. One was English, one was American. I think this is the American here. The English guy was very prim and proper, came to work every day at the same time, took tea at the same time, so forth. The other guy was a little bit different. There's an old story where the English fellow is giving a talk to some scientists about the work they're doing. They're not done yet. But he's giving a talk, and this is in a multi-story building with windows. During the middle of the talk, the American guy rappels down the side of the building in front of the windows where the talk is going on. So I tell my lab students, just because you don't like your lab partner doesn't mean you can't discover DNA, right? Okay, this will be my last slide. Good place to stop too, right? The DNA molecule in detail. Let's look at the numbers first. What was the 0.34 down here? The distance between the bases. The distance between that we call them base pairs. AT is a base pair. Genes are sometimes indicated as how big they are by base pairs. A gene is 1,500 base pairs long, right? 1,500 rungs of the ladder. The twist that goes through, there are two types of twists. There's a big twist and a little twist. And a DNA molecule goes through a big twist and a little twist every 10 base pairs. There it is. Times 10, right? That's what they were seeing there. They were seeing the distances in the twist. The from the edge of the molecule to the first base, okay, so basically we say that a DNA molecule is 2 nanometers wide. Let me change that. It's 2 nanometers wide. 1 nanometer is half its width. Okay, stick with me because I think I'm going to be able to finish it today. All right, not the whole chapter. I'm going to stop on this slide. But anyway, the molecule, again, is anti-parallel. Don't forget that. That becomes very important. What we mean is something called the 5' end. This is the 5' carbon sticks out. On the other one, the 3' end. That's because that carbon right there is called the 3' carbon. And, of course, the space filling model is kind of like what we saw at the beginning, all right? Excellent. All right, I'm going to stop the recording there.